you can. Okay, here we go. So today we're going to talk a little bit about enzymes. We didn't mention that at all yesterday or last time we talked. And the name of these notes is the Enzyme Notes. So that's a little bit weird, right? Let's talk about enzymes. First of all, what are they? Enzymes are proteins. You guys remember we said a protein is a polymer, right? So we went to a nice, easy way to represent a polymer is it's a bunch of repeating molecules. We said it's kind of like a train, isn't it? The whole thing together we call a protein, and each individual ball or molecule in the chain is called an amino acid. Okay, and so that's kind of what they're trying to show right here, right? This is a long chain. You can see there's a bunch of balls there linked together, bonded together. But the thing that I want you to notice is that the protein doesn't stay straight, right? The way I drew it before was it's like a train. Trains are straight, right? They don't kink up on themselves. But in reality, proteins, these amino acids, they are attracted to each other and they repel each other. And so the protein will do what we call protein folding. So let's label these arrows. The protein is going from being linear and it's folding in a sequential way until eventually it's not linear anymore. It's got a three-dimensional shape. It's 3D, right? So it starts off looking kind of like a linear polymer on the left, but then it goes through a bunch of folds, and it folds and it folds, and eventually it becomes a three-dimensional shape. And that's what an enzyme is. Enzymes have three-dimensional shapes, like the molecule on the right side. Okay? And so down here below, this is just another way of representing an enzyme. Okay. And if we could, let's label down here this purple molecule. That's our enzyme. And if it helps, you might even want to take a line and just remind yourself, this is actually all a bunch of linear polymers that have folded onto each other, excuse me, into this very special shape, okay? And this enzyme has a spot on it. I'm going to label it with red just so it pops out a little bit more. This spot right here is called the active site. ACTIVE. The active site. It's a spot on the enzyme that complements the shape of another molecule. Okay, when I say complements, fits together. Kind of like a hand fits in a glove, right? They're the same. They're not the exact same shape, right? My hand is a different shape than my glove, but I can fit my hand into my glove, right? They complement each other. So they fit together at the active site. The molecule that is fitting together with that enzyme is called a substrate. Substrate. You can think of a substrate as kind of like the same thing as a reactant. A substrate is the same thing as a reactant. It's a chemical that's going to be changed. It's going to have a chemical reaction happen to it. Okay? Any questions so far? So we're saying these proteins can fold into three-dimensional shapes that have active sites that fit together with other molecules called substrates. Okay? So we're going to number these because these are meant to show a sequential series of steps here. So first step, second step, third step. In the first step, these guys come together. Okay, so the substrate and the enzyme come together. And in the second step, the enzyme is going to stress the substrate. Okay, so Do you see how the substrate they're trying to show, it started off as yellow, now it's looking yellow and orange? The enzyme is getting stressed. Sorry, the substrate is getting stressed. That means it's getting weakened, right? At the end, in the third step, you can see now that the substrate has been changed, right? It's been chemically changed into two products. Okay, and the products are different than the substrate. That was a chemical reaction that just happened, right? The substrate's bonds broke so that the atoms could rearrange and these products could be formed. Notice that the enzyme did not change. The enzyme was not changed. So the enzyme did not have its bonds break, did not have its atoms rearranged or anything. It stayed the same. Okay, so that means that the enzyme can be reused again for the next substrate that comes its way. So an enzyme is a lot like a tool. Okay, whenever I think about enzymes and what they do in our cells to make changes happen, I think of them as kind of like tools, kind of like a hammer. Okay, a hammer is a lot like an enzyme. It makes reactions or it makes changes happen faster. Makes change happen faster. Have you ever worked with a hammer, maybe pulled a nail out or pounded a nail in with a hammer, right? You guys know if you've done that, it makes putting a nail into wood or taking a nail out of wood way, way faster, right? If I didn't have a hammer, I had to use my hand to try to put that nail into the wood, it would take a long time and I'd probably hurt myself, right? But with the hammer, it makes the job easy, right? Enzymes are like the hammer. Okay, let's label this. The hammer is like an enzyme. It makes a change easy to accomplish. The substrate is the thing that gets changed, right? And so in this case, the substrate is going to be the nail. The substrate gets changed. Right? It's still a nail afterwards, but it was stuck in the wood, right? So that's kind of like our substrate. The substrate gets changed. The hammer stays the same shape before and after the change happens. The last thing to notice is the active site, right? In order for the hammer to affect change on the substrate, they have to fit together, right? And so the spot on the hammer where that's happening right there is what we call the claw of the hammer. That spot on the back is called a claw. That's the active site. Notice that the shape of the enzyme, the shape of the hammer, is not the same as the shape as the substrate, but they fit together. They complement each other. And so that's how the enzyme is able to change the shape of the substrate. Okay, one last thing here. So enzymes control chemical reactions. Okay, they make them happen is what we mean when we say control. They do this by decreasing or lowering the activation energy. You guys remember from Jenna telling us it's the peak, right? The activation energy is the peak on the graph. And so in this graph down here, right, this solid line represents the reaction with a high activation energy. that means the reaction is going to be slow. And that's because there's no tool to help. But the same reaction with the tool, right, starts with the same reactants, ends with the same products, has a low activation energy. Low activation energy means fast reaction. And that happens because you have the right tool. Sure, no problem, Samantha. I'm going to pause here and just zoom out so that hopefully Samantha can see what she needs to see. Okay? Okay, thanks, Ms. Priscilla. Okay, Samantha, Mrs. Priscilla is going to help you with anything that you missed, okay? All right, well, let's just continue on just a little bit more, okay? So we're saying that if we have tools, we get our jobs done faster. It's the same way with the cell. If the cell has the tools, then it can make the change happen quick. But let's imagine for a second that something happens to the tools that changes the shape of the tools, okay? If the tool doesn't have the right shape, can it do the right job? No, it can't. It can't. And so enzymes are sensitive to their environment. If there's a change in pH or a change in the temperature, then that can cause the shape of the molecule to change. Let's imagine our hammer, okay? Let's say our hammer was in a fire. I don't know how that happened, but let's say that the hammer got into a fire, okay? And let's say that the handle burnt off. Is that hammer going to work as well without the handle? It might work a little bit, but it's not going to be as good, right? For pulling nails, you really need a long handle in order for that hammer to work well. And so if we change the shape of our tool, then it doesn't function anymore. Let's say that our hammer fell into a vat of acid. I don't know how that happened, but in my garage, anything's possible. So let's say the hammer fell into a vat of acid, and the metal part, the head of the hammer, let's say it dissolved. So there's no more metal part. It reacted with the acid. And so now all that's left is a wooden stick, the handle. Is that going to work? No, it's not. And so we say when enzymes have the wrong shape, we call them denatured. They're not their natural shape. Denatured means not natural. Okay? And so if an enzyme gets exposed to heat, or if an enzyme gets exposed to acid, its shape changes. And then the end result is that reactions take longer. Why do they take longer? Because the tool is the wrong shape. When something's the wrong shape, we say that it is denatured. Okay? And so I like this little illustration here, right? On the left side, we see an enzyme with the natural shape. And you can see how it fits, right? It's got the right active site so that it will fit that substrate. But then when we heat it, so maybe this is fire or something, or acid or something like that, now the enzyme is denatured. It's got the wrong shape. And that means the reaction isn't going to happen, or it's going to take a long time. I'm just going to jot down no reaction because it's easier to write. No reaction. Okay. That's everything, Ms. Priscilla. Thank you.